Good morning, everyone. Welcome to the March 2025 Zoom Citrus Seminar. Thank you for joining us. Today's program offers one CEU for pesticide license renewal and one CEU for certified crop advisors. If you need CEUs, email me your name, email address, and license number. Today's program will focus on research update on OTC injections. Our guest speaker is Dr. Ute Albrecht, Associate Professor in Plant Physiology at the UF-IFAS Southwest Florida Research and Education Center in Immokalee. As you are aware, Florida citrus acreage and yield have been down. So far, we lost 70% of our citrus acreage and over 90% of the fruit production due to citrus canker, citrus greening, hurricanes, and freezes. But the Florida citrus industry is still very important to our state. It has an economic impact of about $7 billion. We do not have a cure yet for citrus greening, but we have a few strategies and technologies to partially cope with HLB, like nutrition, cover crops, silage management, growing citrus at high density under protective screens or caps, using individual protective covers or IPCs, applying growth regulators such as GA and 2,4-D, and using bactericide, antibacterial therapy, or OTC injections. Dr. Ute Albrecht will summarize the most recent findings from completed and ongoing injection trials. Yield, fruit, and juice quality data will be presented, and the benefits of injections will be discussed. Her presentation will also discuss how to optimize and enhance the efficacy of injections. Dr. Albrecht, please go ahead.
Good morning, everyone, and thank you, Mongi, for the introduction. Okay, as Dr. Zekri said, I will present a research update on OTC injections, and want you to appreciate this nice photograph my student took. He's very artistic, and he's trying to make art out of our all of our injection trials. So, see how I move forward. So, I know there is a lot of different opinions about among growers if OTC injections are really working for them or not. In all, we have a lot of trials going on or completed, and in all our trials, we always find that the injections are effective. And I want to start by talking about some of our trial results, and particularly about some of the rootstock trials where we just completed the harvest after 2 consecutive years of injection. So both of these rootstock trials are with Valencia scion, and this rootstock trial here is at a ridge site near Babson Park. It was planted in 2015. It has 6 rootstocks, 3 of the US varieties, USA 12, 942, and 897, 2 of the UF varieties, UFR 2 and 4, and as a standard Carrizo. We completed the injections in May 2023 and in 2024, also in May. And in addition to just comparing injection, we also compared whether there's a difference if we inject into the rootstock portion of the trunk or into the scion portion of the trunk. And this is a huge trial. These were the trials that were originally set up by CRDF, and so I'm working with them together right now. So this is over 14 acres, and we harvest every year 4,320 trees. So this is a real valid trial in terms of the benefits of injection. And the injections were performed with a commercial injection crew. So here you see some of the data. I have presented the boxes per acre. And on the left side here, the y-axis has all the rootstocks, starting with UFR2 down to US 942. The white bars are for our non-injected controls. The orange bars, the results for scion injection, and blue for rootstock injection. And the x-axis here is from 0 to 300 boxes per acre. And you can see that the results are not overwhelming, but the best producer in this trial has always been US 942. And you can also see after one injection, the yield last year. So it responded rather nicely to the injections, maybe better than some of the other rootstocks, and it was also overall the best performer in this location. So on average, across all the rootstocks, we had 109 boxes per acre for the non-injected trees and 132 to 137 for the injected trees. So that's an increase of 24%. It's not super exciting, but it's nevertheless a statistically significant increase. And as you can see, there may be some fluctuation of whether we inject into the rootstock or the scion, but there is no statistically significant difference. So it really doesn't matter. So let's look at the results from this year. So as you can see, we have the axis in the same way from 0 to 300 boxes per acre. And it's evident that we have almost doubled our production in this site, which was really, really exciting. And you can see that rootstocks that were in the previous year not particularly exciting in terms of productivity, like UFR2, now responded amazingly with more than doubled the yield after injection. You can see here US 897, which is generally considered a more small tree size inducing rootstock, almost at the productivity level of a US 942 or Carrizo. So it was quite exciting to see these data. So across all the rootstocks for the non-injected trees, we had 123 boxes. For the injected trees, 220-ish. And again, there was no difference whether we injected into the rootstock or into the scion. And overall, relative to the non-injected trees, we had an improvement of 79% in yield alone. And as I mentioned before, you can see that overall this trial produced double the number of boxes, which was really, really great. And we need to consider that this trial was also in the path of Hurricane Milton. So we went in the week after the storm and we saw about 30% estimated fruit on the ground. So you can imagine what results we would have had without a hurricane. So here are the results for the total soluble solids. These are only for last year, but the results for this year are similar. Here the boxes in blue are for our non-injected control. Brown is for rootstock and green is for scion. You can see each rootstock responds differently, but they're all responding. And you can also see that US 942 is by far the lowest when it comes in terms of bricks or total soluble solids. And that is something you really need to keep in mind. I know a lot of you are complaining about the bricks and the low bricks and the solids, and it may be because you have 942 in your planting. And it seems to be across all our trials one of the lowest bricks inducing rootstock. However, it makes good looking trees and the trees look healthy. The fruit are large. It just simply means that it needs a longer maturation time before you should harvest it. Regardless, we saw all rootstocks responded. So in addition to your increased production, you also have in this trial, it was a 22% increase in soluble solids, which is also nice. Let's look at the second rootstock trial that we have going on. It's also on the ridge side. It's closer to us here in South Florida. It's near Venus, also planted in 2015. Here we have 5 rootstocks, US 812 and US 942. Again, the 2 UFR rootstocks, 2 and 4, and as our standard sour orange. Here we injected the trees in April 2023 and in May 2024. And the treatments were the same, rootstock injection, scion injection, or no injection. This trial is also a very large, 15 acres, and the trees are planted a little less dense. So we have 3,600 trees that were harvested. And here are the results from last year, for 2024. Again, on the left side, our rootstock, starting with sour orange. And on the x-axis here, boxes per acre from 0 to 200. You can see it's not particularly overwhelming. And this side, we have USA 12 as our most productive rootstock, and it's also the one that responded the most after only one injection. And compared with sour orange, which was one of the worst performers in this trial, really surprising. It was really doing poorly. Across all our rootstocks, we had 49 boxes on average for the non-injected trees and 76 to 80 boxes for the injected trees. Overall, a 59% increase with one injection. And again, it really didn't matter if we injected into the rootstock or into the scion. So how does it look in year 2? So this was this year, and we were again really excited about these data. As you can see, the results are just the boxes per acre more than doubled in this whole location. And if you look at sour orange alone, even in the non-injected state, it more than tripled, it almost quadrupled its production. So it was quite amazing. I don't know what happened, if there's like environmental condition factors, or is it that we're creating a lower inoculum load with our injections in certain areas. So nevertheless, we were really happy about these results. And you can see in the injected states, our orange is now on par with the production of our best performers here, USA 12. And overall, we had 92 boxes per acre for the non-injected trees and an average of 150 to 160 boxes per acre after injection, and which was a relative increase of 70%. But as I said, the controls also improved. So we had, I would hope that at least you made some money out of this trial. Total soluble solids overall much better in this location than in the other location in Babson Park. Still, as you can see, US 942 here too is the lowest PRIX inducer, although all the other rootstocks came maybe close to 10 in the injected state. So overall, it was an increase of 11% in extra TSS. So in some of in every year, we also measured the uptake time of whether we injected into the rootstock or into the scion. Again, brown is rootstock, the bars in green are for the scion. So, and here on the y-axis, we measured the minutes it took after we inserted the injector and opened the valve until it was all taken up. And you can see it took up to 200 minutes. And if we looked at the scion injection, it was significantly faster than if we injected into the rootstock, except for maybe UF-IFAS, which was the fastest in this location with the uptake, regardless of where we injected. So there are some subtle differences, but I will talk about scion and rootstock later in this presentation. So we have a lot of other trials going on. So I want to talk about some of the older trials that we have. One of them is Valencia on Swingle. It's also in Central Florida near Fort Meade. The trees were planted in 2005. So at the time of the injection, they were about 18 years old. We did all kinds of different things in this trial. So one of things we did was we tested different oxytetracycline doses per tree. So ranging from 0.55 and then 0.825, 1.1, and all these were injected with one injector on one side of the tree. And then we used an even higher dose, that's the highest dose that's currently allowed per label, 1.65 gram of OTC per tree. But we split the injection and we injected basically half, so 0.825 on each side. So two-sided injection and the highest dose. But we also tested different months of injection and also different patterns of injection. So we wanted to see if 2 consecutive years of injection are better if we have 1 injection followed by an off year. So we're in our 2nd year, and so next year we will have some more additional results. So let's take a look at the fruit yield. Here I have it expressed in pounds of fruit per tree. And on the left here, you can see the treatments, control, and the different OTC doses, the control in blue. The higher the OTC dose, the darker the color of the bar. And so on average, we had in this location about, 70 something a pound fruit per tree. And when we injected, you can see it really didn't matter at the 0.55 to 1.1 gram dose. So they were all in the range of 26 to 36%. There was really no correlation with the dose, and it was not statistically significant, the differences between the rates. But when we injected 1.65 on 2 sides, we had a significant increase of up to 48%, which was kind of interesting. So looking at the yield from this year, you can see again our control here is just above 50 pounds of fruit per tree. And our injected trees, again, we see, regardless of the injection treatment, we see increases. And again, it varied here based on the dose, but no statistically significant difference based whether we inject 0.55 grams or 1.1 gram. But the highest dose injected with split injections clearly dominated the results here with over 154% increase in production. And don't forget, this trial also was in the path of Hurricane Milton. So and in the week after the storm, we estimated probably about 30% of the fruit were on the ground. So you can see we would have been even better without the hurricane, but unfortunately that's just what it is. So here's just some of the visual of our non-injected trees here on the left in blue, and in right, this is our best performing treatment. You really don't need a map in this trial. You can pick out this treatment just simply by the density of the canopy, and doing the harvest also by the uniformity of the fruit color and size and everything else. So I said we had different pattern of injection, and so the results here again is pounds fruit per tree, and this were the results for this year, but only if we looked at trees that were injected for 2 consecutive years. So these trees were injected in 2023 and in 2024. You can see our control here was about 60 pounds of fruit per tree, and our injected trees, and after 2 consecutive years, and only here I have only the rate up to 1.1 gram. You can see we have 104 pounds of fruit per tree. What happens if we only inject 1 year? So these trees were injected in 2023, but then we did not inject in 2024 and harvested in 2025. You can see again the control the same, obviously 60 pounds. And here we still have a significant improvement in the yield, but it's much lower compared with 2 consecutive years. It's perhaps not surprising, but it's just a confirmation that 2 years in a row are better than only 1 year and then no injection. Let's take a look at the juice quality. These were again for last year. This year they're similar. It's not the grower harvests fairly early in this location, so the pricks is not particularly high overall. But compared with the control, you can see all our injection treatments increase the pricks significantly. Pricks acid, of course, the pound solids per box, and even also the juice color, we see frequently an improvement with the injections. And our best results we always get when we inject more OTC, and in this case, of course, it was a combination of a higher dose and the split injections. So this is always our best treatment. So I also mentioned that in addition to different patterns, different rates, we also tested different months of injection. So if you look at the left table here, this results for 2024. In this year, we compared injection in June and in September. So middle of June, we injected that year, and end of September. And you can see, and in 2025, we made it a little earlier, and I'll tell you later why. So we beginning of June and the beginning of August. So what are the results in 2024 and 2025? We had a significant improvement in yield compared to the control, but it didn't matter whether we injected in June or in September or in June and August. The yield was the same. However, if you look at the pricks, you can see when we inject in June, the pricks is significantly lower than when we injected in September or in August. And this is something that we have observed in other trials as well. But I will get back to this later. So let's take a look first at some other trial that's in the same location, similar location. It's a Hamlin on Carrizo. So also near Fort Meade, also planted in 2005, also 18 years old at the time of injection, of the first injection. So here we injected in May 2023 and in May 24. So that gave us about just the 180 days of the harvest interval to be in compliance with the label. And here we did the same for the Valencia trial. We tested the different OTC rates, again, 0.55 to 1.1, one-sided, and 0.65, two-sided. So here I show the results a little bit differently using the box plots. For both years, we harvested, like I said, in December. So the control, the blue boxes, and the different OTC rates are with the increasing colors indicated here. So you can see in year one, we had a moderate increase after one injection. And so with our best highest dose, we had a 34% increase in yield. If you look at 24, you need to take into consideration that these were Hamlins and they were in the front of Hurricane Milton. So after the week after we went there to assess the fruit drop, so you can imagine more than half of the fruit were on the ground right after Milton. And so basically the top half of the trees were all without fruit and didn't look all that pretty, but we still had some good amount hanging on the trees to get some good data on the yield. And if you compare with the non-injected trees, if you inject 0.55 to 1.1 gram, we had a 75% increase despite the loss from Hurricane Milton. Obviously, we are lower than in the previous year overall, but nevertheless, a relative increase. And if you look at our highest dose, 1.65, administered in 2 split injections, we had a 150% increase. And if you compare the numbers, they're actually the same, like in the previous year without a hurricane. So you can imagine if we wouldn't have lost the fruit, we would have more than doubled probably our yield here. So even Hamlins can respond to the injections. And I know many of you don't measure any, don't think they measure anything, but we did find some significant differences. Also looking at the juice quality, again, all our injected treatments result in an improved pricks, juice color, juice percent, and pound solids, of course. And our best results always with our highest dose administered on 2 opposite sides of the tree. So let's talk about OTC fruit residues. So here are the residue analysis for these both trials, Hamlin and Valencia, that I just talked about. And we analyzed we made a mistake with our highest dose, unfortunately. So these are only the results for 0.55 to 1.1 gram. As you maybe remember, the allowed maximum residue levels in the whole fruit is 10 parts per billion. So that's really a tiny little drop in a big swimming pool, if you want to do a comparison, or 0.01 parts per million. So as if you look at the Hamlins, they were injected in May, harvested in December. So it's just the 6 months between injection and harvest. So you can see the higher the dose that we injected, the higher our residue, which makes sense. but in any case, we stayed 6.9 was the highest, so we stayed below the allowed maximum, so which was good. Looking at the Valencias, and here these are only the results for the June injection. So injection in June, harvest in March, that means we have 9 months in between. And you can see that it really didn't matter what the rate was. We didn't go above 2.5 parts per billion, which is much below what is currently allowed. So which was also, which is good to know. However, as I mentioned, we also injected in September. So here you see the data for June injection is yellow boxes, September injection are the green boxes. Here is the maximum allowed level, so 0.01 ppm or 10 ppb, and you can see that regardless of what rate, even at the low rate, 0.55 gram, September injections led to a way too high fruit residue, despite having an interval of 180 days. So we injected in September, harvested in March, so there was just 6 months. However, because of these results, and we have seen something in some other trials, so it seems to be a consistent trend. September injections are not recommended if you want to remain in compliance with the label and the residue levels. So let's look a little bit more the month of injection. Here you see some photos from this is a trial that one of our earliest trial that we did where we injected some trees here at SWFREC, and we were quite surprised. We injected in spring. That was, I think it was early April, and compared with the controls, you can see my student called it the fruit were comically large. So you can see they're almost double in size, but at harvest time, I mean, granted the harvest was early, it was about February or so, the fruit were still green, and if you look at the inside, the peel is still relatively thick. So needless to say, the pricks wasn't particularly impressive. But when we injected in fall, so in this case, I think it was the beginning of October, you can see the fruit are much better in coloration and peel color here than our control, but they're not increased in size. However, these are our fruit with the best pricks. So this is kind of in line with some other observations from the other trials. Why is this? So we're trying to do some more studies where we do month by month injections. We're starting this year. So I just want to explain a little bit what I think is happening here. So we have the 3 stages of fruit development. I put this together. Of course, it's not quite as simple. Sometimes you have overlapping stages, but basically between March and June, you have the stage 1 or the cell division. Cell division is happening mainly. So the fruits are not very big, but they start to divide the cells. So if you inject during the cell division stage, which is from March to June, so the earlier you inject, you get these larger fruit with the lower pricks, because it seems like we're either stimulating the cell division or maybe we're simply resetting the health and how it used to be. You remember Valencias used to be not harvested in February or beginning of March, but more towards May. So maybe we were just resetting the health, or maybe we're stimulating the cell division. In any case, if you inject early in the year, and I know most of you have the harvest crew here, and then after they're done with the harvest, you get them to inject the trees. So maybe this is one of the reasons why you don't see these satisfactory pricks results in your trials. So keep that in mind. If you can delay your injection a little bit toward May or June, you get much better results. So if you inject, however, during the second stage in fruit development, which is the cell enlargement, so the fruit cell division is completed, all the fruits do is now enlarge. So if you inject here, you won't affect the cell division anymore. And the fruit stay at the same size when you inject, and you will have these smaller fruit, but they're since you're resetting the tree health, you get the higher pricks because the tree is healthy, gets the sugars into the fruit, but the fruit stays small. And of course, smaller fruit in generally have more, I guess it's kind of, if you have a US 897 fruit for example, compared with the US 942 fruit, the US 897 makes smaller fruit with a much better prick, simply because it's a smaller fruit size. So smaller fruit are usually always better in terms of pricks. So anyway, this is something you need to consider when timing the month of injection. And of course, whatever I tell you may be irrelevant because you just need to figure out how to do it logistically. But again, it's something to consider, or maybe start a little experiment on your own if you want to improve your pricks. However, don't forget, don't inject too late. September, not recommended. Like I said, the fruit residues will be too high. We're currently investigating August injections. We completed an experiment and we just sent our samples back to get the data analyzed, but we don't have the data yet. So I hope I have them soon. So I don't know if August injection would be okay. So let's talk a little bit about distribution and sectoring. I know this distribution has come up lately in a lot of conversations, so I want to reiterate what I already talked about in previous presentations. All of you by now will be probably familiar with trees that look like this after the injection. We have one side looking good and one side looking like nothing really happened. So the side that looks better is obviously the side that we injected, and usually it's also the side, if you saw phytotoxicity, even with this is the side that still looks the best afterwards. So we have this typical sectoring if you perform the injections on only one side of the tree. And during harvest, you see that the side of the tree has more fruit with a better color. And we did one experiment where we collected both sides of the tree separately. So the pricks in this experiment was 11.2, and the opposite side only brought a pricks of 9.3 on average. So that's 2 points lower, which is quite impressive. Still, it was better than non-injected trees with 8.5. So to explain what's happening, I want to show some of the dye studies that we did a while ago to explain how things move in the trunk. So here we used a small greenhouse grown tree to demonstrate it better, and we injected it with a pink dye. And here, this picture, this is basic. Then we cut the stem of the tree up at the injection site, and you can see it's this big pink plop at the injection site. And when you cut the trunk just above the injection site, you can see the dye has started to move. You can see these pink spots, but it's mainly still staying on the side of where we injected. As we move further up in the trunk and then in the smaller branches, this is when you can finally see this dispersion of the dye in a circular way inside the trunk. So it will finally, from one side, it will move to the other side and will be more homogeneously be distributed. And then as we get into the smaller branches, it will then concentrate in the sapwood closest to where the under the bark and where the phloem is located. So keep that in mind. And also, if you look at the trees in the field, you can see that most of these trees have a very short trunk. So either it's because the nurseries are crafting lower, or you don't prune the trees anymore after you remove the wraps. So this short trunk is usually not long enough to permit the OTC to evenly distribute in the trunk before it reaches the canopy. So if you have a long trunk and you inject low in the tree, it may have time enough to disperse into the other side, but a short trunk like this, you inject on one side, it will only go in that one side. To show that more visually here, this is a tree that we injected at our center. It doesn't look very healthy, as you can see. Here we hung the injector just a few inches to below the scaffold where the main branches are starting. And after the injection, we injected a pink dye again. You can see everything goes just right into that side of the tree where we injected. So into that branch that is most closely connected to the injection site. Here's a close-up of the leaves. You can see those leaves are all pink. And the leaves on the other side, nothing really went into those leaves. So this is a simple visualization of what's going on with the sectoring when you inject OTC on only one side. Let's take a closer look at the canopy to maybe explain the phytotoxicity that we're also observing. You can see the dye here goes, and some of the leaves will receive a lot of dye, some will receive less dye, and some will receive no dye. So this is why you observe some of these phytotoxicities. In some areas, it's more than the other areas in the canopy, but usually it's on the side that you inject. So you have the sectorized phytotoxicity and the sectorization of yield and health in the tree. And in some cases, if it goes right into a neighboring branch, you can even have really severe phytotoxicity with fruit drop and dieback of the branches, but it's usually only a small sector of the tree. So what can we do to change this? Well, 2 are better than 1. If you remember the Valencia trial results that I presented, granted we had the highest dose, had the best results, but it was also injected with 2 on 2 sides of the tree. So here we have some, we have an experiment going on, and I also maybe urge you to test it yourself. It's really better, instead of injecting only one side of the tree using one, like for example, 1.1 gram, it is much more effective if you split the dose into half, like 0.55, and inject it on both sides of the tree. So that brings us to trunk injury. When we inject our trees, even the older trees, we usually, most of our holes look pretty good year after the injection. You don't see much bark cracking or anything in the tree, and the holes are closed. So it looks pretty good. Sometimes you see a little bit of bark cracking, or sometimes even some worse, but for the most part, when we do our injection ourselves, it's like hundreds of trees instead of tens of thousands, so I understand. But so our trees look usually good. Of course, you can see sometimes this with commercial injections, and these are younger trees, so younger trees are much more prone to bark cracking. And so this is what you want to avoid. And if you choose to try out like double injection or split injections on 2 sides of the trunk, you need to ensure that you're not causing some damage like this, because you may cause more damage in the long run to your tree. So be careful with the injection. Try to monitor the injection crew that they don't jam the injectors in. I haven't figured out exactly what is done differently to see this major damage, but we're working on it. Something that we found, though, when we compared scion with rootstock injection, we found that when you inject into the scion, we have a higher propensity for bark cracking, and usually the bark cracks are also longer. It may make sense because the scion is usually a little bit younger than the rootstock, and in some cases you have a very massive rootstock. So especially if you have young trees, it's always better to inject into the rootstock to prevent maybe the worst trunk injury. But again, we're looking more into this. Also, when you inject young trees, make sure you use the right size injection tip. You can get these 1364 applicator tips with the appropriate drill bit. I also recommend pay attention to your drill bit. Use the ones that are used for wood working. So it's the bread point drill bits. Don't just use any drill bits. Bread point drill bits are designed to drill into wood. Replace your drill bits. Don't inject too deep into the trunk. So just pay attention to what is done to prevent the worst damage. We have 2 formulations that all of you are using. I just call them here OTC 1 and OTC 2. So we have a trial going on where we're comparing both formulations side by side. We have 2 years of data. Here you see the data for yield, fruit weight, and pricks. And you can see in both years, 2024 and 2025, both formulations led to a significant increase in yield, but there was no difference between the 2 materials. Fruit weight was significantly increased. And each year, again, no difference between formulations. And the same for the pricks. Both years, significant increase in pricks. Here was not statistically significant, but nevertheless, no difference between the formulations. And both also cause the same phytotoxicity. I know there's some discussion going on. We inject them, we see the same thing. So no difference. Some words about volume. I know some of you have mentioned that they think if you increase the volume in the injector, you may have a better distribution. So we started a trial last year to test if a larger volume is in fact better than a smaller volume. These were fairly young trees. So we used 0.825 grams of OTC per tree injected into the scion. And we compared a one-sided injection, so either with 50 milliliter or with 100 milliliter, and then compared it with a two-sided injection, again, either with a 50 milliliter or 100 milliliter. So the actual dose was the same, 0.825 grams per tree, but the actual concentration in the diluted solution is different. So just keep that in mind. I know the label talks about ppm, but 100 milliliter of a 8,250 ppm solution will be 0.825 grams. But if you only use 50 milliliter of that, it will only be 0.4125 grams. So it's confusing to talk about ppm instead of the actual dose per tree. So here are the results. It may look complicated, but it's not. I walk you through it. So we have it in two locations. Grove 1, UTC is our untreated control. So you can see we have about 20, this is in metric, so kilogram per tree, so 20 kilogram per tree, so maybe 50 pounds per tree or something, untreated. If we injected the trees with split injections, you can see we have the highest yield, but there was no difference between whether we used a lower 50 ml or 100 milliliter of solution. In Grove 2, the same, our untreated control about 50 pounds or 20 kilograms per tree. Here we see our best results again when we use the dual injection. But we also see a difference here, so that a higher volume is actually better. So 2 injections are better than 1, and a higher volume may be better than a lower volume. So when in doubt, opt for the 100 milliliter instead of the 50 milliliter. Maybe you can improve the distribution in the tree. But just keep in mind that in these trials clearly show that double injections are better than single injections in both locations. So lastly, some words about OTC stability. Also a lot of discussion. By now, you should all know that OTC is not stable once you put it in solution. If you leave it in the lab, like about fairly cool conditions, 72 Fahrenheit, we did it, leave it overnight in the lab, it changes color, it turns orange, but it's still clear. So it's likely still effective, but I still would not store it overnight. But if you mix it and then you fill it in the injectors, and so that's what we did. We filled in the injectors. We laid the injectors underneath the tree overnight. And then you can see the next day we emptied the injectors, and this is how it looked. So a high heat, 80 to 90 Fahrenheit, will degrade the oxytetracycline very quickly. So do not keep it under hot conditions outside. So the heat seems to have a higher impact actually than light. So here again, so this is how it looks, so the lower and the higher concentration. And what we really need is to find something that we can add to the oxytetracycline to prevent its deterioration. And many of you may have heard there's a lot of stuff going on with vitamin C that Brian Updike has come up with the idea to use vitamin C or ascorbic acid as an antioxidant to keep the oxytetracycline stable. So we're having some experiments going on right now. We also have an adjuvant that is developed by Dr. Sandra from UCF, so that can stabilize the OTC. So we're currently looking into that if it has any effect on the tree. If you want to play around, do your own stuff, I recommend you not to do your whole grove, just do a few trees to test things out until we have better data. Because if anything you add to the OTC can cause also some significant unintended side effects like phytotoxicity or so. So lastly, starting over, if you want to continue with citrus production and you want to plant a new trial, I strongly urge you to use the individual protective covers. I know people on the ridge have to remove them usually because of mealybug problems. So here, this is a trial we started last, and we removed the covers. This is actually a Woodstock trial, so we want to go and start injecting those trees. Because usually if you leave them uncovered, if you remove the IPCs like at the end of the season and fall or something, by spring after the flush, they're likely already affected. So if you use a small injector and you do it very carefully, you may, so this is something we want to start. However, the IPCs come in different sizes. If you use these huge TPs here and you remove them, you can see you have these huge trails, and then you can easily go and inject them. And we also have some experiments going on here at our center. This is the two extremes. This was a tree that had IPCs and OTC. And this is the tree that if you don't put IPCs on it, and you don't do any psyllid management, and you don't inject, so it's obviously a striking difference. So path forward, combine the use of IPCs with OTC injections for new plantings, and also always keep in mind that the OTC injections are no cure for all. You still need to do good, take care of your tree. I mean, irrigation, nutrition, and all these things. And obviously you need to continue to manage the psyllid vector, because a re-inoculation will occur even if you eliminated the pathogen with the OTC injection. It's only a temporary effect. And that concludes my presentation. Obviously, a lot of, I have to thank my team for putting a lot of effort into all these trials. We were very busy, and of course the funding agencies, USDA, NIFR, CRDF, and all the grower collaborators. So thank you for listening, and I'll be happy to answer any questions.
Thank you, Dr. Albrecht.